is Carl Ernst. And so, again, just to backtrack a little bit, we realized as we were building this conference that there was a lot of information. And so we were trying to break it down into a little bit more digestible nuggets, as opposed to having it be, one super long session. We know it's a lot of information to absorb, and that's a good thing that this is where we are. So the final one today will be Carl Ernst. We've had Carl speak at our last conference, and so really excited to have an update from him. Him and Eric and I have connected quite frequently over the past year, and excited to have an update today. So welcome, Carl. Thank you for being here.
Thanks for having me, Jeff. Thanks, Eric. Nice to be here. Really nice to listen to everybody speaking today as well. Just show my screen here. Okay, can everybody see that? Everybody see that? Okay.
All good.
Yeah, all good. Great. Maybe I'll get my little more. So I've been working in the Kleefstra Syndrome field for some time now. So my entry point here was working in my postdoc. We were doing a lot of next gen discovery, basically trying to find new mutations for neurodevelopmental diseases. And of course, one of the genes that we hit at the time was EHMT1. So around the same time Dr. Kleefstra was putting out her papers and all this, we were, coming across the same kinds of thing. So I was contacted by a family in Canada, based at McGill University in Canada, after that paper had come out, saying, "Hey, you're working in this area. You've made some models here. Are you interested in getting some of the cells, developing some of the cells, so making stem cells from patients?" So that we can get this going. So the exact kinds of things we're talking about today. So I fit into the cell modeling and therapeutics development. And like I said, it's very nice to hear from everybody today, including Yael that just spoke, because I would be one of those people that sort of fits into one of these modules there. So you can see what it looks like from this particular end and how this would fit into whoever's listening out there, to your personal work on the disease. So what you're looking at here is, these are cells originally from blood. If you just look at the little video in the background, and the cells are converted into stem cells, and then those stem cells are turned into brain cells. And you heard it from Neil this morning, the same kind of idea, but this is a little bit what they can look like. So they're active, they connect with each other. They're a relatively nice way to get a sense of what's going on in what we think is brain-like, without taking any brain samples, obviously, from somebody, and then to study what's going on with a particular mutation in a particular gene and to map the biology. And then, my goal is not to put out scientific papers. That's part of my job, but it's to try and drive discovery and then therapeutics in some of the diseases that we work with. And one of them, of course, is Kleefstra Syndrome. So we began by making these stem cell lines just from 2 patients. And our idea was, let's set this up in 2 cases. One has a small deletion, one has a point mutation in EHMT1. Of course, it's a heterozygous disease, so you still have one functional copy. Presumably, we're going to hear from, I think, Hans tomorrow, where people are going to talk about, that sort of Angelman-like strategy, where are we able to boost the wild-type allele. I'm going to talk about cell modeling and then the small molecule aspect. So you just heard from Yael, that's one way to go. That's one of the more cost-effective routes, but it doesn't target the gene itself. And I'll get to that part at the end of this talk. So I wanted to establish a small cohort of KS models. So we have 2 patients matched to their same-sex siblings. And that's just to make sure that the variation that sort of comes from our systems is as specific as possible to somebody with an EHMT1 mutation. The other way to make sure we're actually studying mutations in EHMT1 is to make it ourselves. And I would say most groups who are in my field, so sort of stem cell biology, neurodevelopmental disease, would do this. And we like this because we take a healthy cell, we induce Kleefstra syndrome in a healthy cell, but we still have the original healthy cells that we can compare it to. So we use this word isogenic, meaning everything's the same. And you don't need to be a neuroscientist to know that the best way to set up a study is to have every single variable identical except for the part you're studying. And in our case, of course, that's a mutation in one copy of EHMT1. So we use something called CRISPR-Cas9. Again, this is a relatively new and efficient technology. And how I like to describe it is it's very accurate, but it's not precise. So we made probably 20 different attempts at knockout, and we get all kinds of different things because the enzyme, the CRISPR-Cas that goes in, does different things to the genome. And here's a good example here. This is the one that worked nicely, and you can see it worked nicely if you look in the middle of the screen here. Oops, can I get my laser pointer? You can see that we have created a Kleefstra-like model here. The control is the original cell line, and the EHMT1 level has dropped about what you would expect to see for Kleefstra syndrome. But how did we make this? This is a picture of the gene. This is the DNA. There's a 29 nucleotide insertion. It means we put something in. This does not occur in Kleefstra. It may, of course, but we haven't seen it yet. And it happens in the first coding exon. Why did we pick this one? Not because it's a perfect mutation that we would see in Kleefstra syndrome, but because it induced this effect here, excuse me. It knocked out one allele by putting in 29 nucleotides and shifting the frame, the reading frame for the protein. So we thought our two patients with their matched controls, the isogenic with the mutant line here, would be a good way to try and discover something about the disease and then to develop these therapeutics around it. So once you have the stem cells, of course, you heard about from Neil this morning, you need to prove that they're stem cells. They need to look like them. That's what you're seeing in A here. This is just from one of our papers. They need to look like baby neurons. So these would be called progenitors, neural progenitor cells, NPCs. We sometimes throw that word around. And then if you keep differentiating them, you can turn them into neurons. And this is not magic. Basically, all we're doing is we're trying to recreate what normally happens in the embryo. We treat the stem cells with molecules that would normally be around when the brain is developing. And lo and behold, if you do it in a dish, they really look and act a lot like neurons. It's not to say that they're perfect, but they're a reasonably good model to get a sense of what a brain-like cell might be like from a real person with a mutation of interest. So that's why we're always talking about this kind of technology. Okay, so if you want to make a therapeutic for a disease, you kind of want to start with what we know about it. And fortunately and you guys have heard about this a lot today, Eric mentioned it in the morning, you're very far ahead on knowing about what this gene does. So the knockout animal, which is usually how people start studying genes, you just completely take it out of an animal, both copies of the gene, and then ask, what does this thing do? What happens to the animal? So the complete knockout of EHMT1, sometimes called GLP, it's the same thing though, is lethal. The animal doesn't survive, and it's got a lot of problems in methylation of histones. I don't have time to get into that for the non-scientists in the crowd, but if you're here, you probably know what that means. I'll just show you an example from this paper from, almost 20 years ago now. This is not from us, of course. This is from the Japanese group that initially made it. And you can see that there's a black band that's disappeared here. That is the EHMT1. And then if you look at the histone methylation, and there's different versions of methylation, you can see that it's decreased. The idea is this is doing something to add this methyl group, and it's got the shape and structure of something that should be adding a methyl group. There's also lots of reports about EHMT1 methylating non-histone proteins, so not in the nucleus. So this was intriguing to us. So we're trying to learn about, where should we study this? So the first thing we wanted to see, this is a full knockout you're seeing on the left, but is this true in Kleefstra syndrome as well? Is it a histone methyltransferase? Can we really detect this? One would expect that we can. So I guess a point of concern for us was that when we actually looked at people's papers in the field, there was not a consistent message on what this looked like. So this is a paper from Dr. Kleefstra, Neil's on this paper, Hans is on this paper. So the sort of Dutch contingent, if you excuse the term. And what they reported was that there was an increase in methylation in Kleefstra syndrome. So now we're talking about the loss of one copy of the gene. So as we've been saying throughout the day, it's really important. What did we just hear from Neil? Good biology, make sure you understand it. This is a point that we, as a community, need to be very certain on. This is a very nice paper. I didn't do it. It just the data look very strong, and yet it's showing us exactly the opposite of what we'd see. Here's a 2021 paper from the people who made the mouse knockout of the Kleefstra mouse, showing us the exact opposite, that the level of H3K9 methylation is dropping. So they cannot both be right. They are definitely different. There's a few differences, but they just can't both be right. Multiple developmental time points here, an 8-week-old brain here. So I think one message I have today is we need to kind of get to the bottom of some of this and maybe decide as a scientific community, we can, of course, everyone's welcome to discussion, what is going on here? And we need to look very carefully at what's happening. So for a lab like mine, we wanted to make very certain of what we were looking at. These are both mouse models. What happens in the human? First thing we did was just ask, can we even see this at all? So we just take a standard drug that is used in anti-cancer therapies and ask, can we pick up any depletion just under normal conditions? And the answer is yes, we can. What you're looking at here is this histomethylation mark. We use 2 different ways to see it, and you can see it really drops. As we add more of the drug, we can really pick up the depletion in methylation. So we are able to see it. Now we can go and test different ways to see if we see the decrease in methylation in EHMT1 syndrome as you would expect. What we chose to do is we said, you know what, let's go to the world expert on histomethylation who's working on cancer methylation. This is what this person does, and this is Ben Garcia at Washington University, and he does mass spectrometry. The only reason that's important, besides being a big fancy word, is that it is a way to check histomethylation without using antibodies. A lot of things we do are dependent on a particular detection system. The nice part about this system, you can look at a lot of things without using antibodies, essentially. So we have our 2 patients and controls. As I mentioned in the beginning, we have our isogenic, and then we made a bunch of replicates of them. So they're the same cell line. We grow again and again and again and again to make sure that we're not just, what we're seeing is not just noise. And then we shipped this to Ben. He was at, I think, Penn State at the time. And he ran it using his own tools and systems. So this data was not generated by me. We just made the cell lines. And what I can tell you, what we saw was pretty much not a lot of methylation of histones taken from neuroprogenitor cells from these from patients and our isogenic control, our isogenic created Kleefstra Syndrome model. The sort of strongest hit was this peculiar one here. This was the top level, and it's barely, this is a barely significant. I know it looks very strong, but this is uncorrected p-value. This is just a straight p-value. There is an increase in it in the Kleefstra syndrome, so it's unclear how to interpret that. It's a particular mark many people are probably not able to study. There's no antibodies to this. I don't know how to interpret that, and I wouldn't say that it's necessarily true. This is just the number one thing that came up. All of the H3K9s were non-significant, so ME2, ME3. What was non-significant, but significant on just a straight p-value test was the ME1, H3K9 ME1. And you can see here the case has a slight depletion of 0.7. Now, these numbers, they look very similar, but the technology that's being used has a remarkable resolution, so it should be seen as not as potentially bad as it looks. So this is 9% versus 8.3%. It is able to detect down to 0.1%, just to give you a sense. But again, this would be considered non-significant. So our conclusion from this kind of has to be, we do not see methylation changes at H3K9 in Kleefstra syndrome human models. And I'm wanting to say this today because we've already have that out there in the literature from the Dutch group. It's at least in mouse, and it would be nice to see more and more of that information being put out where we're looking very carefully at that. To be very certain of this, we did it in a different way. We picked the one that was modestly significant, H3K9 ME1, but didn't pass the significance threshold. And we looked at a region of the genome that the Dutch group had put out where they said they saw the slight increase. And this is kind of gibberish to most people, but what we're looking at is a section of DNA, and you're looking at how much H3K9 methylation we have. Red is a Kleefstra patient, that's case 1 with the about half megabase deletion, and blue is his healthy brother. We did not formally analyze this because we moved on from this, but I can tell you when my eye looks at this, I see higher red peaks. That's exactly what the Dutch group was showing in their mouse model, that there seems to be an increase. So I kind of wanted to emphasize at this conference that we seem to be seeing something similar to the other people modeling EHMT1 syndrome. And like I said, I haven't tested this mathematically, but if your eye just scans across this and you look at the top and the bottom, it looks almost consistent, like the red is a bit higher. This is a specific analysis of a specific region, the one that they had highlighted as important. We're seeing the same thing. I don't quite know what to make of this, but from somebody trying to develop therapeutics, this is not where I want to be putting my, resources of our lab, since there's so much uncertainty around it. And in fact, it seems to be going consistently in the opposite direction that we might expect. The other takeaway from this is we need to be careful that the full knockout is not the heterozygous knockout. So just because it behaves a certain way does not mean that the Kleefstra model is going to behave similarly. It's a very complex interaction with binding partners and how the methylation groups get added. There's different stages of development. I think we need a lot more work on this. So this was just sort of part one of my talk to say this is something we need to look at and probably not a good basis for drug discovery. What I do want to tell you about is we had done this and spent a lot of time looking at the methylation because that's where I assumed that we would go. And when I saw that, it is obviously not where I chose to go because of what I just showed you. So we took a step back, we took our cell models, and we just asked, well, what might be going on that we can actually check? Is there some robust signal that we can see? And we do this with different disorders. I've done this a lot now, and I can tell you this was the strongest effect size we'd ever seen in an initial exploratory analysis. So Neil hinted at this a bit this morning, but what we found, this is an RNA-seq analysis. You're looking at the left. I don't want to get too technical on this talk today. I know there's a lot of parents out there. Essentially what we found was a robust signal of gene expression that appeared to be linked to a single pathway that is well known in all cell types. And it surprised us and it hadn't been reported before to our knowledge. And it might suggest something new for EHMT1 and be the basis for a drug therapeutic development. So this is unpublished data. It's it's been reviewed and it's in major revision, frankly. So we need to do a lot more work on it. But these are parts from this paper that I think, and the reviewers did agree, was quite robust, where we think we have found something pretty unique, important for Kleefstra syndrome that I hope, my goal with this, you heard from Yale that yes, there's therapeutics development, but there's also importance of biomarkers. So I want to get a message out about this system so that as people maybe at Boston Children's or in Erasmus or wherever people are being collected, that this is something that's on the radar and it's something that people can start looking at as potential for biomarkers. I'm not working in blood, but maybe the system does exist in blood. Maybe that can be helpful. So this is sort of the raw data that comes off. There's the case and the control. You can see control 1 and then the case. These are Kleefstra patient that I showed at the beginning. And these are all genes that are important in this pathway called NF-kappa B. And I just wanted you to get a sense of, you keep seeing this increase, and sometimes it's not perfectly significant. Sometimes it's like absent in the control and there's this, it's on in the Kleefstra syndrome, or it's very modest, but here the change is real. Here, maybe I won't go into the RNA-seq, but just to say that it's very consistent across the pathway with a lot of the genes that are known to be activated. And that's why it stood out statistically. Again, these are computer modeling that we do. And this is true. I'm not showing it here, but it's true with the knockout model. Again, maybe I do have that. Okay, I don't, but that's, so we looked across our models and we kept seeing the same thing. So it told us that there may be a hyperactivation of that pathway in Kleefstra syndrome in our brain cell modeling. And this is the model we are thinking is true. And again, I appreciate people aren't necessarily biologists, so I'm going to just try and simplify this. The NF-kappa B pathway activates these series of proteins that go into the nucleus and turn genes on or off. And there's a very complex pathway of regulation. If this pathway is allowed to be active, it's not good for the cell. So it can respond to for example, viruses. A virus comes in, it will trigger this pathway to immediately attack the virus. It's also important in brain development. When we knock out different genes in this pathway, mice memory is affected, the mice have deficits and all kinds of things that could be relevant for Kleefstra syndrome. We think, and we have evidence for, I'm also not showing today, I do know now that EHMT1 interacts with this top part of the pathway. What we think may be happening in Kleefstra syndrome is that this top part of the pathway, which gets activated from outside the cell, EHMT1 functions to keep it locked down and repressed, so it does not activate itself. When you are missing EHMT1, this is also maybe why it's a dosage-sensitive syndrome. There's still some EHMT1 in the KS situation, but there's so much of this complex. Say there's hundreds of this TAC1, TAB2, 3. You only have so much EHMT1 that can bind to it and keep it locked down. In the control state, there's enough EHMT1 to match the level of TAB2, TAB3, this sort of complex, but in Kleefstra, there may not be. So you have some of these that do not have their EHMT1 partner to lock it down, which activates this pathway. You end up with an active NF-kappa B pathway that obviously should not be there. If this happens during brain development, who knows what happens? If it happens in response to a virus, who knows what happens? So it's famously implicated in immune signaling. I'm not trying to make an immune argument here, but if anything, for people who have kids out there, you might expect variable response to viruses or an enhanced protective effect. I'm not saying any of that is true. This is all theoretical modeling. I am in a cell, but that might be a prediction of this particular model. I wouldn't expect any vaccine reactions or so on, but there may be variation in the response, or you might notice that they're getting less coughs than your other kids, for example. Like I said, this is just a speculative on the theoretical model. What is my proof for this? So like I said, I'm not going to spend too much time on all the details today, and we're running short on time, but we have pretty good evidence that in fact it is hyperactive. TNF alpha is a molecule that activates the NF-kappa B pathway. If you look at our control in our case, you can see the HMT1 level is about half of the control. Again, these are real patients. Let's take an output marker of NF-kappa B. Doesn't really matter what this is. We could have picked anything. This one was just expressed in neurons, expressed in MPCs, and had a really great antibody. It's really the only reason we used it. You can see in the control, it is not active. If you look in the Kleefstra case, it is on when it should not be. We have not added drug yet to activate it. So right away, you can see that something's happening that probably should not be happening. This goes with our model of repression of the path. When we add the drug, you can see right away, if you just take the lowest drug, we obviously should have done this at lower doses because we've hit a plateau already, but this was the first time we ran this. You can see that there's a robust increase. So not only is it active, we think, at the non-drug, in the non-TNF case, which is this 0, but as soon as it's added, it's also hyper responsive to any kind of activation. So in other words, EHMT1 is probably tonically repressing it just when it's sort of floating around the cell doing nothing. And when there's a signal to activate the pathway, it's probably not keeping it low enough. And that's sort of the evidence you see here. Now, what if I was to take that drug that I showed you at the very beginning that had the unambiguous effect on depleting methylation? What that drug does is it blocks both EHMT1 and EHMT2, the methyltransferase activity of it. And yes, we picked up the methylation activity. That's not what I'm showing here. What I'm showing here is even in a healthy NPC, if you were to add the drug that blocks EHMT1, so in effect, we are, in a sense, modeling pharmacologically Kleefstra syndrome. Look at this almost dose-dependent increase in this output marker. Even in a very famous marker, I wouldn't call it a marker, but a protein that's relevant for the pathway, you can barely pick it up, but it does seem to be behaving the same way. It's just very poorly expressed in our particular cell model, but I used it because people in mouse have shown it before. So even in a healthy cell, when I add a drug that blocks it, I can see exactly the same type of effect, and it appears to be dose dependent with my drug. The important thing I wanted to talk, so I guess a message here, of course, I'm leaving out a lot of things. I'm hoping this paper will come out. Realistically, it's going to be February or March. We're working on it. And by the way, Neil's an author on that paper too. In the spirit of collaboration and doing a lot of this stuff. We are thinking that this is a potential biomarker. So if you're looking in blood, like please check some NF-kappa B marker, IL-1B, anything. I can, write to me, we can discuss it. The NF-kappa B pathway, I just want to put this out there right now, is a very scary pathway for all drug, Merck and Pfizer and so on. And it's because they've tried a lot of things in this pathway, independent from Kleefstra, independent from brain, and it's very difficult to go in and tackle it. One guy called it a sort of the hairy beast of drug development. So I know that's not nice to hear, but I just wanted to, make sure expectations were reasonable. So it seems to be a very difficult pathway to chase. And for example, there's some drugs that target arthritis and are, I believe, approved that go after this pathway or try to reduce this pathway. Again, a lot of stuff around immunology and that do not necessarily cross the blood-brain barrier. We looked at a few different drugs that are, there's over 800 of these things now, 800 different inhibitors of the pathway. So they're going after different things. And, can I repress this part? If you think back to that map I had at the beginning, we looked at a few of them. most did nothing or did the reverse, or we couldn't pick anything out. But one that we saw behaved exactly as we were hoping for, and that's to reduce the level. Again, if we've lost repression of this path, we want to drug, if we add an inhibitor of the path, we think we might replace what was missing in the Kleefstra syndrome case. When we look at different important output measures, let's just look at the CXCL5, which I showed you here, seems to be a marker of the disease. You can see that this drug is reducing it back in the Kleefstra when we use our highest dose. So that at least was somewhat promising to us. My final slide here, I just want to share with you the patient information that we've used to test this too, and it continues to support a role for this pathway. So the drug we're using, you know, it doesn't cross a blood-brain barrier. It's not approved in human. It's been used before in animals. I don't think it's going to be the be-all and end-all. I'll describe the slide in a second. What I think is that we have an opening here for probably, I think, and I'm very interested to see other people test this, which is why I want to share this today to say, please go look at it. We haven't published it, but look, go check. You know, if I'm right, then you should see the same thing. It shouldn't matter that it's my patients and my knockout. It should have nothing to do, you know, if it's in Canada, so it works. No, it needs to work in, you know, the Netherlands. It needs to work in Japan. It needs to work everywhere. So I think it could be a start point for, you know, is this a relevant pathway? What are the output measures? Are they relevant as a blood biomarker? Maybe, 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 maybe. And what is the molecule that could best chase this? So it's the start point. That's what I'm hoping to introduce you today. Let me give you an example of what this looks like in patient cells that we've made and our knockout. So just follow the vehicle and plus. When we add the drug, you can see every time compared to its control, it gets lower. This is patient 2. Every time we add the drug, it gets lower. In our knockout, every time we add the drug, it gets lower. This NF-kappa B. And when we here, I think this is 48 hours here, we just did the exact same thing. We did it for 24 hours. Your eye can probably pick this out by, you know, by eye, that it's always getting lower in the treated group. When we experimentally induce Kleefstra syndrome, that's this UNC642, and then we add our drug, you can see, I think it's going to be 1 and 2 here. The addition of our inhibitor really lowers it down each time. Now, some caveats here. We're looking at one output marker. This is pretty fresh in the sense that we're really trying to explore what this means. Does it make sense? We need to look at more markers. We need to look more carefully at this pathway. You know, that NF-kappa B, maybe I'll go to my final slide. The NF-kappa B, the TAC1, TAC2, which is where we think is the most important part, it does other things in the cell. So in other words, it could be repressing that pathway, and NF-kappa B is just one piece. So what I'm wanting to share today is just that we think we may be peeling back a new biological function of EHMT1 that might be relevant to Kleefstra syndrome. And at least there's some therapeutics and some small molecules that do what we hope they would do. And now we have a conversation starter with, you know, I think this is still in the academic realm, not yet with the Yales and other people who are ready to advance this to IND. This could all be, who knows what this will end up, but at least it's going to be a start point. So you can see our what we're trying to do here, evaluate more molecules, dig deeper into the outcome measure. That means getting more genes to be like, they should all be behaving similarly. Look at those other pathways that complex interacts with beyond NF-kappa B. And then, of course, the mouse model, we need to check to see if it's doing anything in that. And with that, I'll just thank everybody for their time and being with us at the end of the day. And I'll just acknowledge my student who did all of this work with 2 technicians from my lab. So it's wonderful to see that the students can come on there and do that and just on their own kind of drive something like this forward. So this is essentially his PhD. And of course, I didn't do justice to every last little piece of information. And with that, I guess I will stop. So thank you very much.
Hey, Carl. Really cool presentation. Thank you. I just going to jump in because I had a question. So when Neil was talking earlier about olanzapine and looking at cell models, he had this interesting result that it looks like it's working on astrocytes. And you're working in neural progenitors mostly, or fully differentiated neurons?
So this, we have checked differentiated neurons and can see it, a reduction of the molecule. Our MPCs can give way to astrocytes and oligodendrocytes. So we certainly will look at them, I guess, is my short.
Yeah. That was my question. It's just, yeah, you see where I'm going. It's a really interesting possibility.
Yeah.
What are the key cell types where this may be the most important, this effect?
Yeah, so it exists everywhere. What's possible is that the astrocytes are releasing chemicals like TNF alpha, for example, that are creating the problem, perhaps in Kleefstra syndrome, and that you would need to address every cell type anyway with your, for example, your inhibitor, but that the astrocytes might be the locus of the problem, but the actual root is that you don't have repression of the pathway. So when they're stimulated, you can't get that repression. So these astrocytes are kind of famously involved in some of these inflammatory signals. We keep using inflammatory, but really it's just how it was initially discovered, some of these molecules. It's really, they do a lot of different things beyond that. But I think we've had great support from the Japanese group, actually, with their mouse model, also linking IL-1B increases, microglial increases. So I think it's not a crazy idea that we have here. I think things are pointing in a direction that seems reasonable. Again, Neil has tried this in his own hands and could replicate what we saw. So I'm very hopeful about this, and definitely we will look at different cells. We need to get in the weeds a bit. We need to look at the cell types, Eric. We need to look at the mouse, but at least we know what we're looking for now, and it's like very clear what the hypothesis is. We can easily prove it wrong in the sense that nope, doesn't work, and get rid of it quickly, which is of course what we want to do. Or keep it going if it keeps surviving these tests that we put it through.
Great. Thank you. Great talk.
Yeah, thank you, Carl. I think it's just, to kind of put a bow on the day. It's been really great to see all the speakers be able to kind of weave in and out of each other's talks. And so I think, a massive thank you to all of the speakers, not only for presenting, but also staying present for the other discussions. And we can see how, again, there's a lot of activity taking place. And Eric and I joke a lot that this is a very small world that we're existing in. And you can see how we've had some very fortuitous relationships throughout our 3 years thus far, and it just continues to build on and develop, and seeing the collaboration with the community, within the community, and research as well as our own has been really fantastic. So with that, we will bring today's discussions and presentations to a close. We will begin again tomorrow. If anybody has any questions, comments, concerns, ideas, whatever the case may be, please reach out. I think EIL really drove home the point of what we can do as a patient advocacy community. And it comes around to the worldwide map. It comes down to the data. It comes down to the biorepository, the bio roadshow opportunities. So again, for us to build and get to where we want to go, we often talk about our community being, and our mission being, the three-legged stool, where it has to be the research, it has to be the clinical care, and it has to be the community. And the one thing that we can really control there is our own inputs and our community effort. So excited to see where this takes us over the next couple of days and beyond. Thank you all for joining, and we'll see you tomorrow. Take care.